Good afternoon. My name is Kobie Kirven, and since last summer I've been working on a project studying the effects of increased dietary iron on the functional potential and taxonomic composition of the zebrafish gut microbiome. And I was advised on this project by both Dr. Patuk and Dr. Gordon and the biology department. And then this work was supported by a grant from SC Embry. So here's just an outline of what I'll be talking about today. I'll start with the background and introduction and then move into my hypothesis. That'll be followed by materials and methods, then the results, and this is sort of broken up into two parts. So I have taxonomic analysis and then functional annotations. And then I'll have my conclusions, limitations and future directions, and then my acknowledgments. So what is a microbiome? So you'll hear me use this term a lot today, and you probably heard it on TV or somewhere else. But when we use the term microbiome, we're just talking about the collections of microorganisms and then the environment that they inhabit. And so this environment could really be anywhere. So in this figure, you can see that microbiomes are found in soil, ponds, landfills, and even the ocean and underwater vents. And so in humans and in other vertebrates, we have microbiomes in our nose, in our mouth, and in our gut. And so for this project in particular, I was specifically focused on the gut microbiome. So there are trillions of bacteria living in your gut, but they're not just coming along for the ride. And so you can think of them as being like another organ in your body because they perform many of the biochemical functions that are beneficial for human health. And so this includes the synthesis of amino acids, which are the building blocks of proteins. They also produce B vitamins, including vitamin B1, which is involved in energy production. And then the gut microbiota also produce short-chain fatty acids by breaking down starch. And these short-chain fatty acids provide a lot of the energy for cells within your colon. And so one interesting thing for me about these microorganisms in your gut is how they act kind of like the Avengers, and they protect your gut against invasions from different pathogens. And so this is either by outcompeting them for resources or by attacking them directly. And so one example of what happens when you lose these good bacteria is a C. diff infection, where in many cases of antibiotic treatment, the antibiotics can kill the good bacteria and allow the Clostridium difficile bacteria to take over. And this causes all sorts of problems. And so the microorganisms in your gut do a lot for you, but they can also be affected by many things, including age and diet. And so one of the things that has been shown to change the gut microbiome is increases in dietary iron. And so iron deficiency anemia, or IDE, affects millions of people worldwide, and it's caused by your body not having enough iron for all of its functions. So this leads to a reduction in the levels of red blood cells, which carry oxygen throughout your body, and that can cause extreme tiredness. And so one really popular way of treating IDE around the world, and especially in infants, is through fortification using micronutrient powder. There have been some studies like this one I highlighted here that show that a lot, or around 80% of this iron in these supplements doesn't actually get taken up by the body, and instead it just passes right through the colon. And so one of the consequences of this is that for the bacteria, and especially pathogenic bacteria, iron is a rate-limiting factor for growth. And they've seen that this huge influx in iron can actually lead to higher levels of pathogenic bacteria within the gut. And so this in turn can have all sorts of effects like chronic diarrhea, which can cause death, especially in infants. So as a whole, iron is essential for most organisms throughout the tree of life. So in humans, there's an iron atom at the center of the porphyrin ring in a heme group, and this is the prosthetic group, or the thing that actually carries the oxygen in hemoglobin. And iron is also essential for a lot of bacterial proteins. So here we have a nitrogenase, and this fixes atmospheric nitrogen into ammonia, which is the bioavailable form. And it has 3 iron sulfur clusters that act kind of like wires that allow the electrons to flow. And then an example of a protein that needs iron to function and can be found in both humans and bacteria is succinate dehydrogenase. So this enzyme is involved in energy production. And so as we've seen, iron is essential for many of the biochemical processes, and increased dietary iron has led to increases in pathogenic bacteria in humans. And so the question then became, does increasing dietary iron cause changes to the functional potential of the microbiome? And so when I say functional potential, I just mean the proteins the microbiome can potentially produce, and then all the processes that those proteins are involved in. So this led to our hypothesis that increasing dietary iron levels would caused significant changes in the taxonomic and functional composition of the gut microbiome. And then we used a model organism, Zebrafish or Denio. So as I said, we used the model organism Zebrafish for this experiment, and it has been a popular model organism for a while among geneticists since it's a vertebrate and it's easy to grow. And in recent years, it's also become a popular organism for studying the microbiome. And again, this is because they're easy to grow, and then they're also relatively inexpensive. So we started by feeding the fish normal levels of iron, and normal here just means it's the formulated zebrafish food. And then after 1 week, the zebrafish were split into 2 separate groups. So one group continued to receive the regular fish food, while the other had food that was supplemented with ferrous sulfate. And then after 4 weeks, the gut contents were harvested. And I want to make a note here that this experiment was actually done by Janetta Kishkov, who has now graduated. We were going to repeat it last summer, but we couldn't because of COVID. And so talking about harvesting the gut contents, what does that actually mean? And so here I have a schematic of the entire process once we have our zebrafish ready to be harvested. And so here we have our zebrafish in step 1, and you can see the dissected colon, and then all the little microorganisms that are inside of it. And so just to note, this is not actually the scale here with regard to the bacteria because they're a lot smaller than this, but I just enlarged them for emphasis. And so we extracted all the microorganisms from the colon, and then in step 2, we extracted all the DNA. And so DNA is kind of like the blueprint that cells use to make everything they need to function. And so once the DNA is isolated, it gets chopped up into these tiny little fragments. And so this is where the term shotgun metagenomics comes from, because we're getting the DNA from any cells or organisms that are present in the gut microbiome, not just the bacteria, and then we chop it up into these tiny little fragments. And so these fragments then go to get sequenced in step 4. And so DNA is made up of 4 different nucleotides: adenine, cytosine, thymine, and guanine, or ACTG. And so you can think of these nucleotides kind of like letters in an alphabet. So in the English alphabet, we have 26 letters, and it's the combination of these letters in specific ways that give us words that we associate with meaning. And so in a similar way, the combinations of the alphabet of DNA are what give DNA its function as the code of life. And so sequencing is a way of getting the order of these nucleotides, and it outputs them into a text file, which can actually get really big really quickly. And so just for reference, each sample that we had about 14 gigabytes of sequences, so just a ton and ton of sequences, which we call reads. And so the sequencer does its best at identifying the particular nucleotide at each position, but it can sometimes make mistakes. And so the way that the sequencer accounts for the possibility of mistakes is through something called a quality score. And so this is just a measure of how confident the sequencer is that it chose the right nucleotide. So you can see what that might look like in step 5 here, where we have the sequence quality on the y-axis, and then the position in the read on the x-axis, and all the possible quality scores on the y-axis. So we can use that information to trim our reads so that we only retain high-quality ones. And to do this, we use something called quality filtering and trimming, and so I used a program called BBDuck. And so the next step, step 6, is to filter out all the zebrafish DNA. And so like I said earlier, shotgun metagenomics doesn't care who the DNA belongs to, which is different than other methods like 16S. So we want to make sure that we get all the DNA from the bacteria and other microorganisms in the gut that are not the host. So to do this, I align each of the reads to the zebrafish genome using a tool called Bowtie2. And if they matched up, then they were considered to have come from the zebrafish themselves. So if they didn't align, then we knew that at least they were something else besides zebrafish. So then when we had our filtered sequences, it was time to do the actual analysis, and that's in the purple over here. So I'll just briefly go through each one of these, and I'll talk more about them later on. But I started by doing taxonomic analysis, and that's step 7 here. And so this is essentially figuring out what species of organisms are present, and then if they change in the experimental. And so the next step I did was differential abundance analysis in step 8. And so this is looking to see if the levels of these taxonomic groups change between the normal and the experimental. And then finally in step 9, I looked at the functional potential of the microbiome, which is what the microbiome is capable of doing biochemically. And so this purple section is really where I'll be focused on for the rest of this talk. So the first thing I did in the taxonomic analysis portion was look at alpha diversity, and they used the vegan R package for this. So alpha diversity is just a measure of species richness, and that just means how many different types of species are present in the sample and then in what levels. So what does that look like? On the left here, I have a simple example of how we calculate alpha diversity using the inverse Simpson metric. So each shape here represents a different species. And then R here is the richness, and it just means how many different species we have. Capital N is the total number of observations. And on the right here, we've summed all of the number of observations for each species. So then we can plug those values into the inverse Simpson formula to get our diversity value, which in this example would be 4.56. And so I should note here that alpha diversity doesn't tell you who is there, it just tells you how diverse each sample is. So if I changed each one of these species but kept them at the same levels, you would get the exact same alpha diversity. And so on the right here, you see a box and whisker plot showing the alpha diversities, and they're stratified by treatment group. And so when I compared the two groups using a Wilkinson's rank sum test, I found that there were no significant differences in alpha diversities between treatment groups. And so the next diversity metric I looked at was beta diversity. And so unlike alpha diversity, beta diversity does care about who is there. And so this is a way of seeing how closely related two samples are to each other. And so here I use the Bray-Curtis dissimilarity measure. And so again, I have a simple example here showing what that calculation looks like. So here, the S is the total number of observations in the sample. And then for each species that share between the two samples, we take the species count of the lesser one. So in the case of the blue triangles here, there are 3 samples, 3 in sample 1 and 4 in sample 2. So we take the count from sample 1. And then we can plug those values into the Bray-Curtis formula to get our beta diversity. So now this number is going to be between 0 and 1, where 0 means that the two samples are compositionally the same. And so I did this Bray-Curtis dissimilarity calculation for each sample, compared with every other sample. And then on the right here, you see a visualization of that dissimilarity in a principal coordinate analysis plot. And so this essentially tries to reduce that high dimensional matrix into an XY plane. And so here, the closer 2 points are to one another, the more similar they are to one another. And so I then did an analysis of similarity, which is a non-parametric way of testing whether we can reject the null hypothesis that the similarity between groups is greater than or equal to the similarity between samples within groups. And so the results of the analysis of similarity tests were not significant, and so the null hypothesis was retained. And this capital R value right here is just an indication of similarity. So the positive number shows that there was more similarity between samples within the groups rather than between the groups themselves. And so what does this mean? This means that dietary iron did not significantly change who is there in the microbiome. So like I said, I didn't see any changes in who were there, but then the question became, do the abundances of who is actually there increase or decrease after we increase dietary iron? And so just for reference, organisms are classified in this hierarchical system where the most broad is domain, and then you keep getting more specific as you go down to the species level. So I first looked at differential abundance at the phylum level, and so on the x-axis here, you have the class of the bacteria, and then on the y-axis is the log-fold change, which is a measure of how the bacterial levels change in the experimental. And so these are only significantly different ones, meaning that they had a p-value of less than 0.05, and they're ranked from left to right on the most significant p-value. And so the dots here, the color-coded dots, are based on the phylum that they belong to. So here, a negative value on the y-axis would indicate that there's a decrease in that particular group in the experimental, whereas a positive value will indicate an increase in the experimental group. And so overall, the phyla that appeared to have the most significant increase in the high iron group were the Firmicutes and the Bacteroidetes. And now protobacteria were also significant here, but there were some that were increased and some that were decreased. So right now I'm just going to focus on the Firmicutes and Bacteroidetes. And so zooming in on just the Firmicutes, I saw the classes that had the most significant increases in abundance were the Bacilli and Clostridia. And so Bacilli are known to be beneficial to the gut microbiome in humans, and they're in a lot of probiotics. And this is because they can essentially help to drown out potential pathogens. And then Clostridia are also beneficial bacterial group because they produce a lot of the energy metabolites for your colon. And so for all in all, for the firmicutes, our data showed that increased dietary iron increased the beneficial bacteria. And so that was not the case for the Bacteroidetes. And so zooming in on the Bacteroidetes phylum, I found that the most significant changes occurred in the class Flavobacteria and specifically Flavobacteriaceae. And so I started looking in the literature about Flavobacteriaceae family, and I found that its members are actually pathogens of freshwater fish. And so in the figure to the right, you can see examples of freshwater fish that have Flavobacteriaceae infections. And so A and B are both rainbow trout, and then C is the Chinook salmon. And so like I said earlier, previous studies have shown that increases in dietary iron increase pathogen abundance in humans. And so it's interesting to also see this pathogen increase in the zebrafish after we increased dietary iron. And so the last thing I looked at was the functional potential. And this is essentially the biochemical potential of the gut microbiome. So as we talked about earlier, the microbiome is metabolically important for human health. And we're just seeing what the microbiome is capable of producing and if that changes when we increase dietary iron. And so over the summer, I did an assembly-based approach and then looked at what proteins the microbiome could produce that way. And then this semester, I decided to take an assembly-free approach and look at annotating the reads directly without assembly. And so to do this, I use a program called MyPhaser. And what it does is it takes the reads, it translates them, and then compares them to their manually curated database of enzymes. And so it then outputs the enzyme commission numbers along with the number of reads that corresponded to each one. And so I normalized the counts based on the sequencing depth for each sample and normalized them to counts per million. So I then used a machine learning program called Weka with the InfoGain attribute evaluator algorithm and then the ranker search method. And this calculates the relative worth of each enzyme commission ID by measuring the information gained with respect to experimental group. And so then I picked the top 15 ranking enzyme commission numbers and then annotated them using the KEGG database. And so KEGG it's a database that has different pathways that these enzymes are involved in. And so this figure on the left here, you can see a heat map where the samples are on the x-axis and then the top 15 most informative ECs are on the y-axis. And then the darker the blue here indicates the higher the count of that specific gene. And so I also looked at the number of unique ECs that MyPhaser identified between the two groups. And I did a Wilkinson rank sum test that showed that there were significantly more unique functions in the normal iron group versus the high iron group. But just note here that there was a relatively high interquartile range in the normal samples versus the high iron group. And so I know this table right here is a lot, but here we just have the 15 most informative ECs along with their enzyme class, the pathway that they're involved in, and then the median normalized counts here on the far right. And so note that not all of them had associated pathways, so in some cases I just have the enzyme name. And I'll be focusing on a few of these for the next slides. And so the highest ranking EC from Weka was 1.3.1.98, and this is also known as Merby. And so Merby is an enzyme that's involved in peptidoglycan synthesis. And so peptidoglycan is this structural component of bacteria. And gram-negative bacteria have a thin layer of peptidoglycan, whereas gram-positive have a thicker layer. And you can just think of them as like chains of sugar. And so certain pathogenic bacteria like Mycobacterium tuberculosis are known to modulate their peptidoglycan synthesis in a way that helps them maintain pathogenicity. And so during periods of dormancy, they reduce peptidoglycan synthesis and use those sugars to produce energy for the cell. And now I'm not sure why we saw potential decreases in peptidoglycan synthesis here, since differential abundance appeared to show a lot of growth. But I saw a similar thing happen with another pathway. So from the functional potential, I saw that there was a reduction in genes for enzymes related to purine metabolism, and specifically those involved in purine synthesis. And so the reaction of EC 2.1.22 or per-N is the one that I've highlighted here. And so what's interesting about purine metabolism is that studies have shown the gene for the per transcription factor is negatively regulated by the iron-dependent repressor protein FER. And so you can see what that looks like in the figure to the right here. So here, the iron bound to FER prevents the transcription of the per gene, but if the per gene were to be transcribed, it would produce this per repressor protein that then acts as a negative regulator for purine biosynthesis genes. And so this shifts metabolism from degradative to synthetic, and this generates the purines needed for DNA replication and other energy needs associated with growth. So like with Merby, the reduction in these genes associated with purine synthesis would indicate dormancy of the bacteria and not really growth, which again is sort of contrary to what we saw in the differential abundance data. So from my analysis, I didn't see any significant changes in alpha diversity, so species richness, as well as I didn't see any changes in beta diversity, so who is actually there. I did see increases in the level of firmicutes and bacteroidetes after increasing dietary iron, with the firmicutes being those beneficial bacteria, and then the bacteroidetes had that pathogenic Flavobacteriaceae. And then in sort of contrary to the increased abundances, we saw decreases in genes for enzymes associated with peptidoglycan synthesis, as well as purine metabolism, which together would indicate dormancy rather than growth. And so this seems to be telling two different stories, right? One of possible bacterial growth, and then another one of dormancy. So with this functional potential, it's just means that the genes could be transcribed, not actually that they necessarily are. But still, I don't really know how to reconcile these differences. And so some of the limitations from our experiment were that we had a small number of samples, so it was difficult to do statistical tests. And then, like I said earlier, we're looking at functional potential versus transcription, so we don't know if those genes are actually being transcribed or not. And then we're limited by our databases, right? So not all the reads were actually able to be taxonomically classified or functionally annotated, and this can mean that we're missing parts of the puzzle. But really, we just had to work with what we have. And so for future directions with this data set, I think it would be cool to do network analysis, and so this would essentially link together the functional data with the taxonomic data. And then I would like to do another experiment with varying iron levels so that we can do correlation analysis. And then if money wasn't an option, I would like to increase sample size because it would give us more the ability to do more robust statistics. And then also I think it would be cool to look at RNA-seq data. And so this would tell you the genes that are actually being transcribed and not just the ones that are present. And then also metabolite data would give us another measure into the metabolic activity of the microbiome. And so we could combine the metabolite data, the RNA-seq data, and the taxonomic data, again, to do a network analysis and sort of combine all of those together. And so all the code that I used to analyze the data set from start to finish is available on GitHub. And then these are my references. So I'd like to thank Dr. Gordon and Dr. Batuk for their guidance throughout this project, and for Dr. Batuk putting up with me using the supercomputer this summer. And then I'd like to thank Presbyterian College for this opportunity, SC Embry for the funding opportunity, and then Janetta Kitchkov for doing the actual experimentation. Thank you to Rob Highweiler for helping me get set up on the supercomputer, Mr. DNA for handling all of our sequencing needs, and then let's just thank our microbes in our gut because they do a lot for us. Thank you.